I realize that I'm the only obstacle between you and lunch, so I will try to make it entertaining. At the same time, I'm recording this, as you can see here, and I will put this in the web after, so you can, if you sleep out of hypoglycemia, then you can check it out in the web after. First of all, I would like to thank the organizers for inviting me. I notice a twofold enrichment factor on the number of females in the audience as opposed to speakers, and this is good and bad. So I'll look at the good side. The good side is that I expect in 5 years the majority of you giving the lectures instead of all guys like me. Okay? So keep up the good work and become PIs, and you can do it. All right? I will be speaking about systems pharmacology, and I was given the Beyond the Pill thing. I didn't know it was something that is a repeating concept. I saw that there are 2 or more lectures called Beyond the Pill, so I have my own interpretation of what they meant by Beyond the Pill. I will be speaking about systems pharmacology, which is many things, and in my case it's the interaction between macromolecules, which are targets or multiple targets, with the drugs, the modulators of this activity of these drugs, and this is done via their interactions through structures. Of course, everything in nature is interactions at the human level as well as the molecular level. Without interactions, there is no life. And the interactions are what modulates, and to understand interactions, you need the structures. At the same time, we have to understand everything from a systems level, which is complicated because we don't know all the data yet. So what do I feel that Beyond the Pill could mean? Well, some of you may be too young to have seen The Matrix. It's a story of a drug dealer called Morpheus who's trying to subvert an innocent guy called Neo, and he offers him a red pill or a blue pill. The red pill is knowledge, the blue pill is ignorance, which is a bliss, and we're very ignorant in drug design, which has been a bliss because there are many drugs that would not have been discovered, would not work if we had to know the target yet. In any case, system pharmacology for me, Beyond the Pill, is actually combining the two and having better drugs through knowledge. But this is not a very new thing. If you heard of Sidney Brenner, Nobel Prize, he is said to have had in his door a sign that said that the only science with omics in the name is economics. So what is really all the omics noise around? And isn't this just physiology? It is, but it's physiology for the 21st century. It's physiology where we understand really what's happening at the molecular level. So that's where systems understood from structures and from the whole functioning of a cell can help. So you have 2 different models, a more traditional model in which, more traditional in the modern sense model, in which you have a target and you screen and you try to test and find drugs. But you have what was traditional became out passé, and now it's back in fashion, which is to do a phenotypic screen. Forget about anything. Try to get a drug that affects your condition in real people, hopefully in animals first, but in the past it was in real people. And then try to figure out why it works and how to improve it. So what I mean by systems pharmacology is connecting these 2 sides of the coin. Another slide of philosophical rambling. two more famous people, Richard Feynman and Alan Turing. Richard Feynman had in his blackboard the phrase, "What I cannot create, I do not understand." He was very smart. He also had know how to solve every problem ever solved. He could do it. That to me signifies, when I translate it to biology, that only when we can truly develop computational methods to simulate biological systems to the level that they are indistinguishable from the experiment, we will be able to say that we understand biology to the same level as our technology to understand biology allows. This is similar to what Alan Turing created, which is the Turing test, in which if you cannot tell the difference if a system, which is a human or a computer, is answering your questions, you have a truly intelligent system to the best of your questions, which in our case are the experiments, for example. So the two things are related, and you see a lot of biological talks where you see ovals and pentagons that interact, that inhibit one, activates the other, and all this magic. There is so much biology hidden behind these ovals and pentagons that only when we understand it from a structural point of view at the molecular level, we will really uncover the biology of it. So coming back to down to earth, after rambling, in this picture here you can see each dot is a protein, and two proteins are connected by a line if they both interact with the same set of, at least, I think in this case, 10 small molecules. Okay? So each of these 10 small molecules interact with two proteins if they are connected. So each one of these 10 molecules could inhibit the other protein. This is a huge mess just to understand how cells, which of course are do not have all these proteins at the same time, but a large section of them could be interacting with a particular drug. It could be interacting with many targets. This is both a problem and an opportunity, as our previous speaker mentioned, that we can repurpose drugs. At the same time, we have problems with cross-reactivity, with side effects of drugs. I will be speaking about these things. But more specifically, for example, here you see all the yellow targets, I think, are for asthma, if I'm not mistaken. And that means that maybe your target for asthma is not druggable. In this case, these are. You cannot hear the microphone?
Il vient de lâcher. Est-ce que la batterie est encore fonctionnelle?
No, it died. Should I stop the lecture for now, or?
No, you can continue. No, continue.
Okay. Can you hear me still? I can speak a little louder. I have a 4-year-old. I often have, not because I shout at him, but often I have to speak to my wife with a high level of background noise. So here you have a lot of asthma targets, and maybe not the case of these ones, because all of these actually have a small molecule that do interact with one of them. But you could have other targets that the one specifically you're interested in is not druggable. But by means of this network, the evolution and I have to use this? Okay. By means of, as a result of evolution and as a result of the basic scaffolds that repeat themselves and molecular interactions, you can often find a different target that maybe is druggable and could be an interesting target for your project, that could, instead of the one that is too difficult. Of course, molecular interactions and evolution are mixed together, and I should be using the pointer, but when I look at the screen, the camera stopped tracking me, so you can see it was supposed to move. but it's not. Yes. I'm sorry, this is the first time I'm trying this. In cells or in biology, you can have a target that is promiscuous as the ones that we saw before, that binds to several small molecules, here represented by the triangle, the circle, or the square. This actually I'm representing not just small molecules, but potentially different reactions. So there's also reaction promiscuity in cells. But then through a process of divergent evolution, this protein A will have mutations that will make it specific either to one particular reaction or one particular ligand. And this is a process of divergent evolution. At the same time, you can have convergent evolution where different proteins evolve independently to have the same function, which here I will use it in a broad sense, for example, binding ATP. In the human proteome, every other protein and their cousin and friends bind ATP. So if your drug is an ATP analog or a purine-containing drug, it could be very complicated. But you can exploit these things, either to find a different target or to use a different molecule. Now, more specifically to two aspects of systems pharmacology, one from a genome scale, cellular modeling, and afterwards a second application on structural side of things. I will talk a little more of what we do in our lab. Here you see a map of a famous map of all the reactions ever in a human cell, I think. Or is it all biology? I don't know. But what you have to take home from this image is that this is not a real cell. Not a single cell has all of these reactions at the same time. Okay? But let's suppose that it was. What we do with metabolic network analysis or metabolic simulations is that we take that as a factory and we have input metabolites and then we have output metabolites plus a new cell. So we try to simulate how a cell reproduces. And you have a reaction, which is a biomass reaction, everything that a cell needs to live, and everything that it produces, which is ADP, hydrogen, phosphate, and biomass, which is a new cell. Imagine that as a single reaction. And then when you look at this reaction, you have the input metabolites in green, you have the output in some shade of blue. I'm not sure what color is it. They say that men only have a 16 color palette. And then you can inhibit a particular reaction and see if it prevents the production of something that was essential. So that target is something that is essential. In the case of an antibiotic, it's a good target for a new antibiotic. Other reactions you can find different ways. For example, we live here in Montreal, which is an island, has a few bridges. If you break all the bridges, we're stuck. We still have an airport though, but cells don't have an airport. One of the most recent applications was in the case of diabetes. So remember that big map of all the reactions? Actually, we had to build the actual network for a human adipocyte, which is the cell that stores fat in its lipid droplets, and it's related to diabetes. When I'll come to that in the next slide, I think. So we had to build this network, and 95% of the work was in cleaning out garbage in the literature. So our previous speaker spoke about databases. There is so much noise in these databases that a lot of the effort in bioinformatics is to get a clean data set and trustable data. But at the end of the day, we had a network that has 1,300 approximately genes. Of course, these genes produce their enzymes, and these enzymes are involved in about 5,000 reactions with almost 2,000 unique metabolites involved. What do we do then? We take this network and if we knew the reaction speed of all these enzymes, we could make a an uber perfect simulation, but we don't know. Often in systems biology, in computational systems biology, you have two approaches. One is a single pathway where you know all the reaction constants and all the information, and you make very precise simulations. The other is metabolic flux balance analysis, in which you find the subset of speeds for each reaction that optimizes the output of the whole network. Okay? These are not the real reaction speeds, it's just the values that if you will plug on each of the reaction, it maximizes the output of the creation of a new cell. Okay? But then we can go and mutate each one of these enzymes and see how much it decreases. With a mutated model, with a single or multiple enzymes inhibited, we can find what's the best rate, and we can compare how much is the best rate of the mutant with the best rate of the wild type. Okay. In the case of the adipocyte, we didn't want to kill cells, of course. We don't want to kill humans, but we wanted to prevent the storage of lipids. So if you're someone like me that enjoys a beer way too often, you have a lot of fat, and it's better that it gets stored in more adipocytes than larger adipocytes. I'll come back to that. So our idea is to prevent the adipocytes to grow but not kill the adipocytes. We had to find out what it means to kill a human cell. So there is this data of I think CRISPR, if I'm not sure, where every single human enzyme in our case was deleted and knocked out, and checked if the cell was still viable. So we can do the computational work and try to find what threshold of decreasing the biomass production when you mutate a protein that is supposed experimentally to lead to maintain a viable cell still maintains a computational cell viable. Like that, we find the threshold of 0.8 fraction of wild type biomass production as the one that maximizes the agreement of what we find that decreases the growth of a in silico cell compared to a real cell. So, as I mentioned, you have many adipocytes, or you have larger adipocytes, so hyperplasia and hypertrophy. And when you have larger adipocytes, hypertrophy, this is a pre-diabetic condition. So if we can find a drug that would force your system to produce more adipocytes rather than larger ones, this could prevent diabetes, the onset of diabetes. So this is another aspect of beyond the pill. Here we have preventive medicine in the game. Like I said, we take our network, we find the biomass production, and we make mutants, and we compare all of that. So in this graph here, you have on the x-axis the ratio of wild-type lipid droplet production, and on the y-axis the ratio of wild-type biomass production. Everything above 0.8 on the y-axis is what we said that it's supposed to not kill a human cell. So we want everything on the upper part on the y-axis. And on the ratio of wild-type droplet production, we wanted everything that has a considerable effect in decreasing the ratio, the size of the lipid droplets. So everything in the purple or yellow quadrants for the lipid droplet is okay. Everything on the purple and pink for the y-axis is okay. So the union of this, the intersection of these two is the purple axis, and there we find a number of targets, and these targets, some of them are known to affect lipid droplet and some are new. And we went on in collaboration with people at Laval at the University of Sherbrooke to test if obese, diabetic, and non-diabetic people had increased levels of these enzymes as a small indication that this may be involved in these processes, and at least for a few of these targets, in particular FAD, what is it? FAD 1 and 2, I think. I cannot see even my own slides, I need new glasses. We found statistically significant differences in the correlation. So obviously the next step is to take these potential new targets and start using them. So the second application of systems pharmacology was with C. difficile infections, where we wanted to find, in this case, the essential targets. And long story short, we made a reaction, and we proposed a number of essential targets. And a year later, someone went on and mutated every single gene in C. difficile and found which are essential or not. And there are 50 targets that we found that are in agreement with experimental, which in terms of being essential and potential new antibiotic targets. It's a bit cheating here to say that we have 89 accuracy, but I mean most genes are not essential, so yeah, it's easy to have a good agreement on the non-essential ones, and we missed about half of the essential ones. But still, for a fully computational technique, it's quite impressive. Now for the second part of the lecture, I'm not sure how long I have been going on.
You have 10 minutes.
10 minutes? I can do it. I can always do it. So I will touch here on the question of similarity, and you have different ways of measuring similarity. Here I'm talking about similarity from the point of view of the binding sites, not similarity from the point of view of small molecules, as was mentioned before. And you can go all the way from very simple representations of what something is similar, so sequence, strings, or annotations, all the way to interaction energies. And you can go from a local representation, from binding sites, to a global representation, so you can have sequence comparisons or fold similarities. What you can use that for, you can use to predict the function of proteins. You can perform, you can create biased libraries based on similarity, detection of cross reactivity of targets, side effect of drug, understanding the side effect of drugs, drug repurposing, or polypharmacology, which, if you haven't heard, means hitting two or more targets with the same drug, which has some benefits. In particular, patients are more likely to take a single drug than two drugs. So methods for the detection of similarity, there are many. Isoclef was one that I developed when I was a postdoc with Janet Thornton in Cambridge. There are a few others that are older or newer. They all differ in how you represent the surface of the protein or the interactions, how you search for similarities, and how you score. By the way, these 3 points, representation, search, and scoring, apply for any optimization problem, even in your own personal life. You gotta find a way to represent a problem, how to find for solutions, and how to score them. So this is true for everything. Now, what I will be mentioning here is using molecular interaction field similarities. So instead of looking at the surface of a protein, we look at the void within the binding site where a ligand would be there interacting with the protein. And we create a grid, and we use chemical probes and a scoring function to develop sort of a pharmacophore model. So each point in that grid with a different color means that a particular chemical probe is the best, it's the most preferred one in that position to interact with the protein. Once we create that for a pair of proteins, forget about the protein and any ligand that may be there, we have a cloud of colorful points, and the task is to find the largest subset in common between the two. For that we use a technique called graph matching. I will not get into the details, but it allows us to find the largest subset in common that can be actually superimposed between the two. These two proteins have less than 15% sequence identity. They have different enzymatic roles, different EC numbers, but of course it's a bit of cheating. This was a training example. We knew in advance that they bind the same ligand, but they do. And not only do they bind the same ligand, they bind the same ligand utilizing the same interactions that are similar between the two. So if you actually superimpose the two proteins based on the similarity, the two ligands are almost perfectly superimposed. This is not a docking simulation. This is just superimposing the two proteins. So if your target was the blue and your drug was that molecule, it binds to the green one. If you didn't check that before spending $10 million in your drug developing project, you are in trouble, or you could be in trouble. The method that we developed called ISONIF is freely available, by the way, and it's the best performing at the moment, or was in 2015, I guess, when we published. Hopefully it's no longer the best, meaning that we want things to advance. And let me give you some examples about it. So here first we have a kinase which is CDK1 or 2, I'm not sure. And we look at similarities against a large data set of human proteins with known structure. And among those there are, of course, other kinases, but also, and they are spread all over the human kinome, for example. Most often in drug design, companies would test only a few of the similar kinases based on sequence, but as you can see here, the target is here in the AGC branch, but you have very similar kinases in terms of the binding site all over the kinome. If you were to ignore those, either you have no effect or you can have drastic side effects. Not only that, we have other proteins which have very important sounding names like synapsin, and your drug could be interacting with that protein, for example, if it binds in the same position in the binding site. So this sort of detection of binding site similarities can be very useful to prevent side effects if you consider them during your drug development process. Here for repurposing, this is Celebrex, COX-2 inhibitor. And when you look at the binding site similarities of COX-2, among the top most similar proteins binding sites, you have carbonic anhydrase, and carbonic anhydrase was eventually found to be a target of Celebrex, and it's a drug, it's a cancer target. Another example of the use of this technique, rationalizing the side effects of drugs. For example, torcetrapib, this drug is a target for CETP. It was eventually dropped out after 15 years and almost $1 billion because it causes hypertension. And you look at the binding site similarities of the primary target of this drug and our structural proteome data set. Among the top targets, you have several proteins that are involved in the RAS system that leads to hypertension. So had this been checked before, this drug could have been created with slight modifications that would prevent binding to these other proteins, for example, and maybe prevent the side effect. So if they had given me maybe 1% of what they spent in this project, this talk will be in the web. Maybe they'll hear it and still give me the money. This is an interesting case of captopril. Captopril is an approved drug, and it has several side effects. And here what we did was to compare the binding sites again of the target of captopril to our structural proteome data set, and then look at the most similar proteins if they had associations with diseases or conditions that have similar similar behaviors, the word is escaping my mind, than what is seen as side effects of the drug. Okay, so for example, the first target here, it's involved with leukopenia, thrombocytopenia, and the target that is not the primary target of the drug, but is similar to the binding site of the target of the drug, and that is one of the side effects of captopril. Another one, alopecia, which is the hair loss, and there is one of the similar targets is associated with a condition that produces alopecia, and that's a side effect of the drug. And a third one, again, this is more difficult to rationalize because the word cardiovascular incidence at the molecular level is very broad, but still you have a target that leads to that and is very similar to the binding site of the drug. In the case of these three potential cross-reactivity targets, we then went on to use our docking method, which I'm not speaking today, maybe SSM12. These drugs, captopril, bind to these potential cross-reactivity targets in the same position as it binds in the target based on the similarity of the interactions. So all the interactions that are found similar between the target of captopril and the cross-reactivity targets are the ones that the drug interacts with. So this is an independent corroboration of the hypothesis that this drug, captopril, could bind to these other targets. Of course, these are all hypotheses and remain to be tested in the lab. So, as a conclusion, computational systems biology can help identify novel or alternative targets. It can help in terms of the systems biology. And when you involve a structural aspect to it, it can help you understand side effects. It can help you repurpose drugs and go beyond the pill. So with that, I will thank the members of my group. I will thank the past members of the group, the funding agencies, and as a small anecdote, I have two students who in the past attended SSM in Brazil, and that led to a paper after. So that's a retombe, a benefit of the school. So thank you very much for the effort.
Thank you very much, Rafael. It's very interesting talk. So you're exhausted, you're very hungry. You have questions or no?
Okay, no, very interesting talk. So you produce arrangements of points with.
It's a pharmacophore model.
Yeah, but can you produce virtual molecules out of them? If you have a very complex, not efficient,
We
pharmacoscope, but can you get just
Yeah, so
real or virtual molecules.
right. So there are methods that would take a pharmacophore model and try to, in a sense, align real molecules with the atoms that favor those type of interactions in those positions in space. More traditional methods, well, actually, different docking methods actually just go brute force, but we don't have methods at the moment to do that, but they exist.
Very interesting talk, definitely.
Thank
I
you.
have a question regarding the design or the theoretical design of your cell models, the design,
Yeah.
and the application for that in therapeutics.
Yeah.
If I understood properly, you design the model, and then it's with your all your metabolic networks, and then you can identify a number of genes you can hit in order, for instance, to reduce hypertrophy. And that's good in terms of therapeutic treatment. The question is, can you somehow identify genes that by hitting them would result in some sort of metabolic reprogramming and even changes in the phenotype of the cell? Because, for instance, in diabetes, I imagine browning is something that you would like to get, and there are already known effectors for browning.
Yeah.
But could your model be applied to such a situation in which ultimately you're moving from your divisional model, which is an adipocyte?
Right. This is the frontier research in computational systems biology, in the sense of building higher and higher representations of the cell with additional networks. So gene expression and so forth. So at the moment, the data is not available to the extent that it exists for metabolic networks, but in smaller scales there are cases, and even for very small cells, such as, it escapes me the name, but the organism that causes gonorrhea. There are whole cell models of the entire, everything, every gene, every reaction, every function, and then you can simulate those, but also the methods are still evolving to do that more precisely. But this is the frontier. I cannot do this at the moment with this model for sure.
Okay, so thank you very much, Rafael, once again.
Thank you.